This brings us to our next big idea, the origin of life. Put forward in the 1920s was a hypothesis called the Oparin-Haldane hypothesis. This was forwarded by two men, a Russian here, Alexander Ivanovich Oparin, and John Burdon Sanderson Haldane. This kind of looks like a guy that would eat all the magpies and cats in the neighborhood. Their hypothesis posited that life on Earth could have arisen by a step-by-step chemical process from abiotic material, non-living matter, via chemical evolution. A gradual chemical change led to the formation of life. Now, this became known as the primordial soup hypothesis. These scientists suggested that life began in a strongly reduced environment. In this case, the atmosphere was strongly reduced, and the atmosphere chemically exchanging with the ocean, resulting in these life-forming reactions taking place dominantly in the ocean. Ultimately, they were sparked or started by reactions of these reducing compounds with sunlight and lightning, and the net result was a suite of simple organic molecules. This strongly reduced atmosphere is an atmosphere that was proposed to be rich in hydrogen-containing gases, such as methane and ammonia. However, as we'll see, these two gases are sensitive to sunlight. This led in the 1950s to the Miller-Urey experiment. You may remember Urey's name. He discovered isotopes in the 1920s. He continued doing crazy important stuff until the end of his life. In 1953, while he was at the University of Chicago, Stanley Miller, one of his graduate students, worked with him filling a flask with gases that they thought would represent this early reducing atmosphere. These included hydrogen, methane, ammonia, and water. And then they charged it with electricity. So they put some conductors here within this vessel that contained the reducing gases, and they ran water through this whole system, seawater. As this water moved through the system and passed through this chamber where the sparks were combining, a brown slime appeared on the inside of the apparatus. This brown slime was found to contain complex organic molecules, and among them were amino acids that serve as the basis, the building blocks for proteins. Proteins are composed of one or more chains of these amino acids, and they are useful as transport molecules, enzymes, and hormones in modern life forms. This experiment changed the world. It was super exciting, caused an enormous amount of controversy because now suddenly God wasn't required to create life. And now over the years, it's gradually been reconsidered, principally because of the sensitivity of ammonia and methane to light. The incoming radiation, solar radiation, lyses or splits these molecules, and therefore they would have been fairly rare in the atmosphere. So we've gone from the proposed prebiotic atmosphere, a reducing atmosphere, to our biotic atmosphere today that consists of 20% oxygen. These questions, the questions about ammonia and methane and sensitivity to these early environmental conditions, have led to a new way of looking at the origin of life. This new way of looking at life is known as RNA world. Thomas Cech and Sidney Altman won a Nobel Prize in the late 1980s for this, and principally it was for the discovery that RNA molecules, specific RNA molecules, could self-cleave. And if they can self-cleave, they can replicate themselves. They unzip and they fit a new zipper to the unzip section, and therefore they've recreated themselves. This is important because RNA molecules can do this on their own. DNA, on the other hand, a more complex molecule, requires an enzyme, and this enzyme is made of proteins. So this implies that our current DNA world was preceded by an RNA world. We can't replicate DNA without proteins. Those proteins have to be made by something, in this case by RNA. This RNA world is a proposed stage, a hypothetical stage in the evolution of life on Earth in which self-replicating RNA molecules proliferated before the evolution of DNA and the subsequent proteins. RNA-based life might not have been the first life to exist, and alternative chemical origins have been proposed by a number of scientists. This is ongoing. Evidence for an RNA world is significant enough that the hypothesis is generally accepted by scientists. The formation of all 4 RNA building blocks has further strengthened this hypothesis. This is an abiotic generation of these building blocks. Like DNA, RNA can store and replicate genetic information. This information can include proteins, RNA enzymes, which are also known as ribozymes. These can catalyze, they can start and accelerate chemical reactions that are critical for life. One of the most important critical components of the cells, the ribosome, is composed primarily of RNA. This is another strong indicator that RNA world is a good idea. So this RNA world requires formation of bases such as adenine, guanine, cytosine, and uracil. Ribose, C5H10O5, is composed of 5 formaldehyde molecules that could have been created in a CO2-rich atmosphere by reaction with sunlight. So merely bouncing sunlight off of formaldehyde molecules could result in the formation of ribose. This formaldehyde is soluble in rainwater, therefore soluble in seawater. Ultimately, it would all end up in the ocean where it can spontaneously form sugars. Now, we form the other bases starting with adenine, C5H5N5. This can be synthesized from 5 hydrogen cyanide molecules, which are the chemical HCN. So it's a bit odd that things such as formaldehyde and hydrogen cyanide may have led to the formation of early life, even though for us today, they're incredibly toxic. One of the issues with this theory is coming up with sufficient cyanide molecules. The reason we're worried about that is that methane is a precursor required, but methane is readily destroyed by sunlight. So we have to have another way to generate methane other than in the atmosphere. It turns out methane is emitted from mid-ocean ridges. This is a byproduct of serpentinization, in which seawater reacts with ultramafic rocks, forming the mineral serpentinite. This is a methane seep off the coast of Oregon. It is only vaguely related to this hypothesis here, but it's a good picture of methane seeping out of the seafloor. Other theories of life's origin. Many researchers conclude that the hydrogen cyanide and formaldehyde molecules would be too few and far between to ever meet up. They also argue that they would be destroyed by incoming solar radiation and heat. So this requires you think about some other way to get to this point in the evolution of life. What else are we thinking? Well, one of the things that's been considered is interplanetary dust particles, or IPDs. These are organic molecules, including amino acids, that are recovered from IPDs and certain meteorites that have been found in interstellar dust clouds. If these molecules survived the collapse of the pre-solar nebula, they could have arrived on the many bombs that hit the Earth between 4.5 and 3.8 billion years ago. Most of those bombs coming between 4 billion and 3.9 billion years ago. Hydrothermal vents present a third possibility for the origin of life. Water passing through a kilometer of seafloor gets heated, picks up a whole bunch of dissolved materials, and then rises through a ridge vent, through a black smoker or a white smoker, most probably. While passing through the crust and the sediment, the water is going to pick up some key ingredients such as reduced hydrogen, H2S, and ferrous iron, which is going to be soluble in the hot anoxic water. Black smokers occur when 350-degree water hits cold seawater. It generates a dark plume of predominantly iron sulfide, FeS, as well as many other compounds in lesser amounts. So these are fine particulate minerals that are being produced in great abundance. This material rises with the hot water. As that water cools, that material settles back down to the seafloor, where it can serve a number of important purposes. These vents are rich in the reduced materials needed. They have plenty of liquid-solid interfaces like pyrite to help organize the organics. Because the temperatures are too high to form and preserve organics for any length of time, the next step probably took place off-axis. So we have to get away from the actual heat source in cooler water where these chemical reactions can produce the appropriate materials, and those materials are not quickly denatured by the hot water. The result is a spectacular biosphere that's fed entirely by chemosynthesis. The next question is, well, when did this happen? Up until the 1840s, it was believed that life evolved at the beginning of the Cambrian period. This is when we see animals. This is the explosion of life, the evolutionary Big Bang at about 543 million years ago. In the 1940s, microfossils were discovered in very old rocks, much older than the 543 million year old Cambrian rocks. And in some instances, single-celled organisms were discovered in siliceous rocks. The apex chert fossils in the Warrawoona Formation have been dated at about 3.5 billion years ago. If they are fossils and not just carbon chains from hydrothermal vents, then this represents our earliest record of life. Stromatolites, on the other hand, are common in slightly younger rocks around the world. Additionally, it's been proposed that the low carbon isotope values in 3.85 billion year old Isua rocks of West Greenland were indicators of life. And that gets back to life preferring carbon-12 over carbon-13. But it turns out this probably isn't true because some of these rocks are known to be of an igneous source, therefore never had anything to do with life. At the base of the universal tree of life, sequencing of RNA and DNA has permitted the development of the history of increasingly complex organisms. And one of the important breakthroughs that has led to this is the development of what's called PCR. You've probably heard of this. It's used in crime investigations, amongst other things. And it allows for the development of a tree of life with three main branches, starting out with very, very little material. That's the key. PCR can take a little bit of material and make a bunch of it, enough so that we can analyze it and evaluate it for a variety of scientific purposes. We know that bacteria and archaea are single-celled organisms we call bacteria. Eukarya, or the eukaryotes, can be single-celled organisms as well as all higher plants and animals, like us. This is a diagram with some photos that illustrates some of these potential early life forms. They look like life forms, simple life forms, but you can also generate fairly similar looking things abiotically. That remains an issue. And we can run all the way back to the beginning of life using PCR and little bits and pieces of organic matter that we obtain from a variety of living forms today. It's possible that hyperthermophilic bacteria, these are bacteria that live above 80 degrees centigrade, were potentially responsible for this beginning of life. And this is because they're all found near base of the tree of life. What's the significance? This is Grand Prismatic Hot Spring in Yellowstone Park. Huge and beautifully colorful. The color is basically a thermal indicator. Each color represents a different water temperature. The purple, dark blue is the hottest water, close to boiling and sometimes boiling, fading into blue-green, and then green, then oranges, yellows, and browns. So cooler water here, still warm or hot to the touch. Very hot here. I mentioned earlier in class that a guy went hot potting in a hot spring a couple years ago and he dissolved quickly. He dissolved before the rangers could get back to him. This relationship between hyperthermophilic bacteria and these hydrothermal waters may represent an origin of life scenario, or it could be a coincidence where the greater molecular stability of the guanine-cytosine bonds found in the DNA would just make it more likely to persist or survive. So just so happens that the bacteria we find in these hot environments have the genetic makeup that they do because that genetic makeup is stable at those high temperatures. It doesn't have anything to do potentially with the origin of life. So this is where the primordial soup thickens. Life could have evolved in surface waters during the bombardment period in Earth's history and then spread to all environments. Shortly afterwards, impacts would sterilize the land and surface waters. This is during a heavy bombardment period, with only the very deepest water organisms surviving to eventually colonize the Earth's environments. A new idea is that viruses may be involved. Viruses use both RNA and DNA as genetic material. RNA world is currently favored because RNA molecules have the ability and in general have more diverse functions than proteins. RNA can be a genetic molecule, as in some viruses. It can be epigenetically controlled. It can epigenetically control the expression of genes like microRNA molecules. This is big in medical research these days, using this kind of control to influence resistance to diseases. It can have enzymatic abilities like ribozymes and many others. Proteins, on the other hand, are mostly known as enzymes. It seems that an RNA molecule has a better chance of catalyzing its own replication because in theory it can do that. Proteins are good enzymes, but they can't serve as genetic material, and therefore they can't make copies of themselves. That's a major problem. Even though it looks like RNA molecules seem more likely, arguments between scientists continue.